Apologize for that. Um, so again, I'd like to welcome you to our 2024 annual update report on the Stone Research Foundation's research progress. Every year, Dr. Stone and I give a live webinar after the release of our 2024 annual report and provide highlights of our findings in the past year and the upcoming studies, and of course, end with a Q&A session and give you an opportunity to ask any questions that you may have on our research, current, or what we will be discussing today. And this doesn't mean, of course, that this has to end right here. This is just a start, and I welcome any questions or concerns or suggestions that you may have following the webinar. You may email me directly or any of our members of the team. This year, the theme for our webinar is, of course, regeneration. And as some of you or most of you are familiar with our work, know that we are doing our best using and taking advantage of regenerative medicine, the new science and techniques that are becoming available to us day after day as research progresses. And we try to utilize that in the orthopedic sciences and, of course, in the future for the orthopedics. So moving forward, I would like to give an introduction to our foundation and what we are basically persevering every day to do. Our vision at the Stone Research Foundation is to accelerate healing and to treat, prevent, and eventually cure arthritis. I would like to introduce you to some of our key members of the team. On the executive team, of course, our founder, chairman, and CEO, Dr. Kevin Stone, who's here, of course, with me. Myself, our director of communications, Dave Hopkins, and our director of development, Vicki Weiland, who's also present on this call, and you will be hearing from following the webinar. And of course, I'd like to give a shout out to our research and development team here, Simran, Dylan, and Mia Liku, who are key folks here who make this happen every day with their super enthusiasm, intelligence, and hard work that we are progressing forward and pushing our science forward. And of course, again, a thank you to all our research interns who are very active and help on many of the other activities here at the foundation. In line with that, I'd like to thank and a shout out to our scientific partners, collaborators, whom, again, I would like to remind you that science does not happen with one or two people. It takes a team, and it definitely takes a team here at the foundation to basically advance our science from one to the next step. I'd like to give an overview and just a highlight to the main collaborators, some of whom are present here with us today on this webinar. And I would like to start with Dr. Anthony Ratcliffe, who is the CEO of Synthesome in San Diego, one of our main collaborators on the project that we just completed, in fact, and followed by Dr. David Frisby, who is a world-renowned equine surgeon at Colorado State, whom we will be starting our collaboration and work in upcoming months. Dr. Daniel Randy, of course, at Feinstein Institute in New York, who is one of the key collaborators whom a lot of our discovery stage and in vitro studies are done in his laboratory, and we do a lot of our work there before moving on to the more advanced stages in our pipeline. Dr. Julie Rosser, who is, of course, in Austria, CEO of Regeneration Inc., who has a very cool technology called Lab on a Chip, and we have been involved with her for the past few years. And again, that is all in our collaborative wide network globally that we work with. Dr. Mark DeCoster in Louisiana State, who has a very interesting and exciting technology using a copper nanoparticle that, again, we are trying to utilize to help regenerate cartilage cells. Dr. Yuri Galili here in Chicago, of course, is a CSO of Meridius, which is a biotech in Toronto, Canada, that we have just forged a relationship with. And of course, the CEO, co-CEOs of that company, I believe, are present with us today on this call. And on that note, I would like to move to our next slide. That wasn't me. Okay, here I would like to give you an overview of what today's webinar will entail. So basically, there are two streams, if you will, to our research. There is a non-surgical therapies that we are actively working on using regenerative medicine. and injections, of course, and I will get into that later on in my overview. And then, of course, articular cartilage paste graft technique, which Dr. Stone started successfully over 3 decades ago with very good success. And that's something else that we are currently working on right now to basically get to the 2.0 version of it and augment it to the next level, again, taking advantage of regenerative medicine and techniques that now we have available to us. And then, of course, we will hear from one of our patients, Will Hendricks, who received the paste graft technique many years ago as a testimonial as to how successful this technique can be for cartilage regeneration. And then following that, of course, I will go over, again, at high level, the current research that we are doing right now for cartilage regeneration, a little bit more details on that. Following that, of course, I will walk you through how we disseminate the information that we find here. And then we will hear from one of our executive board members, who is one of our main advocates and supporters of the foundation, Mark Bailey. And then, of course, I'll open the floor then to Q&A, and then Dr. Stone and I can answer any questions that you may have. So the non-surgical therapies, I will start with this again. What are we talking about when we say non-surgical therapies? Our hope is one day, and I know that Dr. Stone's hope is one day, that you don't have to undergo the knife necessarily, and unless it's absolutely essential. So this means the injection therapies that some of you have heard of, PRP being one that I'm sure is a familiar name to most people, is being utilized right now in most clinics, including here at the Stone Clinic, with good success. What is the aim for these therapies? Well, right now, it's obviously to reduce pain, inflammation, and eventually, we would like to promote cartilage regeneration. We have an orthobiologics study, which basically is the first of its kind head-to-head comparison, all of the main commercially available orthobiologic injections that some of the clinics are offering both in the US and abroad, to determine which one of these biologic injections reduces the symptoms for cartilage damage and degeneration better, which one is more potent, and then which one is the one that we can move forward with as a therapy to promote cartilage preservation and regeneration. So let me give you a little bit of background here on what these orthobiologics are. So there are different parts of the body that we can source these orthobiologics from. And some of these areas that we are using in, or rather sources of growth factors that we're using in our study, are the synovial fluid-derived MSCs, mesenchymal stromal cells. These are, of course, the support cells in your body that go to the site of injury and release the growth factors that are necessary for healing. And one of them, of course, this is basically the fluid that's the knee lubricant fluid in your knees. That's the synovial fluid. Adipose-derived cells, or the growth factors from adipose-derived MSCs, which are basically your fat cells. So a lot of clinics are now doing liposuction and using that to extract these growth factors from these cells and inject it back into you. Platelet-rich plasma, this is PRP. I've already talked about this. Most people know what that is. Of course, that's taking your blood, spinning it down, and giving you the platelet-rich plasma back to you, again, with the rich growth factors that are pro-healing. Bone marrow-derived MSCs, and again, you've heard of this, I'm sure, has come up. That's another source. And exosomes, which might be not so familiar of a term to you, but these are basically, at best, one could say, packages of helpful molecules is what we call them, that they transport these good growth factors to the site of injury, where they are needed in case of trauma or any kind of a damage. And Wharton's jelly, which is sourced from donated umbilical cord after birth and or C-sections. And it's a very rich sort of population of cells called Wharton's jelly. And again, this is another source of growth factors that is utilized for anti-inflammation and or pain. And we will be using this as one of the arms in our study. Next is, of course, the surgical sort of side of our research, and that is on the articular cartilage paste graft. And on that note, of course, I'm going to turn this to Dr. Stone to give you an overview for this technique, and then we will go from there. Dr. Stone.
Unmuted now. So thanks so much to all of you for joining us. And Monty, great job for an overview. And I hope this webinar is helpful for you to understand our passion around trying to regrow cartilage and regenerate the tissues that all of us go out and injure every day, unfortunately, in one sport or another. So articular cartilage is that shiny white surface on the end of the chicken leg when you crack open the chicken. That white shiny surface, that is the bearing surface of everyone's joints. And when you damage that surface, it doesn't generally heal well on its own. So you can have an acute lesion where you damage it through sport, or you can have arthritis where it develops wear and tear over time. So we developed a technique in 1991 to regrow that articular cartilage. And we did it in the following steps. First, we realized that injuries to cartilage that are not big enough to stimulate a healing response don't heal on their own. And so the technique that we were building upon, the microfracture technique popularized in the 1980s, generated a repair response by making small holes in the articular cartilage. But the repair response was only fibrous tissue and it didn't last long. So we came along in 1991 and said, listen, when we look inside someone's knee, especially when we take away the intercondylar notch where the ACL goes through the middle of the knee, we noticed it always regrew. That was number one. Number two, we knew that there were marrow cells that seemed to produce bone healing and probably cartilage healing. We didn't call them stem cells back then. We also knew that in the articular cartilage, there were signaling factors that told those marrow cells, hey, become cartilage when they come to the surface and when motion is applied. And so we took these lessons and created a paste of articular cartilage and bone, did a super microfracture called a morselization of the damaged area inside the joint, whether it was arthritic or an isolated chondral defect. And then we put the paste back into that defect and applied motion using a CPM machine or a continuous passive motion machine. And then we followed these patients over time. And we realized that about 1/3 of them developed articular cartilage indistinguishable from normal as read by an outside musculoskeletal pathologist. 1/3 would develop a mixture of fibrocartilage and 1/3 would develop fibrous tissue. So over the years, we wanted to improve that articular cartilage technique. And that's what we at the foundation now have been studying hard. Do we add stem cells to it? Do we add a hydrogel to it? Are there other factors, growth factors that we could add to it? With the goal of seeing if we couldn't make articular cartilage paste grafting a much more popular and successful technique for repairing arthritis or arthritic defects. So here in this next slide, I'd like to introduce one of the patients that we did early on. And here he was a young boy, and we'll pop to a slide, as a young boy at 14 years old, he had what many kids have called an osteochondritis dissecans lesion. And that means within his knee, there's a dead area of articular cartilage. It happens to young people, not just older people with arthritis. And the traditional surgery would be that a surgeon would go in and either take the piece out or fix it with little darts, screwing it back to the bone and hoping that it would heal. And unfortunately, many times they don't heal because the cartilage and the bone underneath is dead. So we did an articular cartilage paste graft for him in 2006. And then a second look arthroscopy looking at that damaged area and how well it grew a year later. And he did spectacularly well, returning to soccer and essentially doing back to normal activities. We'll let him tell his story here in a minute. But the idea was that we could take dead cartilage and regrow it. And our job now is to do that routinely, to do that even better than we could ever do it before. Go ahead. Oh.
Okay, I'm going to see if
Hang on just a sec. We'll get Will's voice on there for you in just a sec.
My name is Will Hendricks. I had an articular bone pace graft procedure when I was 15 years old, which was about 18 years ago. It was a really challenging time in my life, just like I'm sure a lot of the patients who arrive at Dr. Stone. So I was pursuing a competitive ice hockey career. I had played hockey and competitive sports my whole life. To make a long story a little shorter, I ended up having a knee injury, significant impact to my knee. I got hit during a practice, slid into the boards. My knee pad had slid down, and it kind of created a bad impact. It was a really dark time in my life because I was someone who had defined myself as an athlete forever. We're referred to Dr. Stone. My mom and I hopped on a plane from the East Coast and came out here and met him, and it ended up being a good fit. I was a good candidate for the surgery at the time. Um, my understanding is I was probably one of the youngest people to have the operation done as a 15-year-old, but we decided it was the right thing to do. And fast forward to today, I just came in for my 18-year checkup, and Dr. Stone looked at my knee and he said he wouldn't even know that the knee was operated on or artificially repaired based on looking at it today. I have no limitations. I have full range of motion. I can do whatever I want physically. I did not continue pursuing that career in sports, but I've continued to play sports my entire life because this experience has been such a positive impact on my life, my family's life, my ability to continue moving forward.
Okay, great. So on that note, after hearing from Will, I like to move on to the current research that we are doing now, as I mentioned before, on cartilage regeneration. So in partnership with and support from the Stem Cell Agency here in California, California Institute for Regenerative Medicine, we received a sizable grant 2 years ago to start our investigation and our study on what we've been talking about so far and what Dr. Stone has been alluding to in improving the pacecraft technique using and utilizing the new science that's now available to us. And this, of course, involves the new, what we call preparation method and how we, in the OR, will be preparing the cartilage and adding the biologic additives, such as the hydrogel, which is basically a glue, if you will, to the paste so that it can stay in place better. And other additives, of course, as growth factors, such as the ones that I just discussed earlier in the orthobiologics. In line with that and in parallel, we are now about to start a large equine study that we are doing in collaboration with Colorado State University with Dr. Dave Frisby. And that work is actually about to start on December 4th, as a matter of fact, where we are looking into this technique in the horse model for this defect of this injury for cartilage regeneration. And hoping that, of course, we can, by doing this, bring this treatment in addition to the human side of clinic to our equine and canine patients as well on the veterinary side of the clinic. The ultimate goal, again, is that this will be a one-step low-cost solution that will be globally accessible for cartilage regeneration. Here, I'd like to go through this schematic diagram. I know it's busy, but bear with me. I will make it simplified by just going through it with you. Basically, starting on the left side, I want to show you where we've started and completed our work from the preliminary research side to the Stem Cell Agency-funded discovery stage project, where we have now enough data and encouraging enough to proceed to the next stage, which is the translational stage, which is basically in the middle of in the dish and the clinic side. So that's where the translational stage research is. And that's where we are at right now. It's pending, of course, applications to the funding agencies and our fundraising efforts to be able to enable this work to push it forward. In doing so, again, as I alluded to that in my previous slide, we're not just doing this for the humankind, we're also actually, our intention is to benefit that in the veterinary side as well, so that the canines and the equines and potentially other animals that do suffer from cartilage degeneration can benefit from this as well. All of this is, of course, under the guidance of the FDA and supervision of them from here moving forward. And without their approval, of course, we cannot get to the clinic. So it's best to seek their advice and suggestions as we take every step from here on forward. Now, in line with that, again, Dr. Stone and I do travel nationally, internationally, to meet with key thought leaders in the field that we do need to consult with. We need their suggestions, we need their advice and their expertise to help us basically excel to the next level. And that is, as an example here, I'm going to give you is one of the meetings that Dr. Stone attended in September, where he presented on our orthobiologics injection study. And I said that we are about to embark on the next study, but what I didn't say is that we already have finished one study that we completed a few years ago. And based on that, we have presented our data now and are presenting internationally and nationally at different conferences so that we can both spread the word, if you will, of our research and our results, but also seek advice for the next studies and how we can forge collaborations with others. I traveled to Copenhagen and I attended a stem cell meeting in October. And there I also met with the head of the FDA's cell biology and cell therapy. And we are now in conversations with them and engaging with them so that we are, again, as I said, under their supervision and with their guidance moving forward every step so that we are doing everything as effectively and efficiently as possible, again, to the clinic. Part of our mission here at the foundation is the teaching mission. So that is, of course, to teach not just the interns and or the residents and or our of course associates here, but to spread and disseminate the research that we're doing and the results that we are obtaining globally. And in doing that, of course, we do it through different channels. We do this through peer-reviewed publications. We do it through conferences, as I said, that we attend, presentations and posters, and through teaching of the postgraduates, fellows, and the interns that we actively host here at the foundation. Now, in that, I originally mentioned that one of our executive board members and champions for the foundation, Mark Bailey, is present here with us, and I'm going to open the floor to him, of course, to share a few words. Mark?
Thank you, Monty. I'm impressed by the number of people in this call. It's awesome. I'm, as Monty mentioned, a member of the board of SRF, and also my wife and I are on the leadership council. We found Stone Clinic years ago when my wife Laura, who was a former Ironman athlete, was experiencing, and she played ultimate frisbee years ago, and she injured her left knee, and she got to the point where she was having more and more problems with the knee, and we decided that she needed to do something about it, and so we went to the Stone Research Foundation or the clinic for a partial knee replacement. And she had an amazing outcome. So she's now back to playing tennis. She's active. We're so grateful for that. I found my way to the Stone clinic just a few years later when I had I ended up going over the handlebars of my bike and and I needed my shoulder fixed up. And I'm also very active too. I do biking, I do karate, and I enjoy being active. Both of us were drawn to the clinic, I think, because of the emphasis on excellence and also the emphasis on preserving the organic function of the body as much as possible. We both have had great outcomes, and I think when we both arrived at the clinic, the thing that impressed us the most was that the play forever mentality, the pictures of the athletes all over the office, the sense of Dr. Stone saying, what's your end game? What's your goal? And that was in high contrast to other doctor's visits where you sometimes feel like you're sent to the principal's office. What'd you do now? And we celebrate and are incredibly passionate about the mission of Play Forever. We think that life is movement, and if people can move and stay in motion, there's all kinds of social benefits, personal benefits, and all that requires joint health, and it requires pushing the edge of research to figure out how we can repair joint issues when the joints are injured, how we can prevent the injuries from happening in the first place, and how we can continue to keep people mobile and active into our later years. It's the term health span has been made popular by a book called Outlive by Peter Tia. And the notion is, as the years roll by, what matters most is not our lifespan, but our health span. Can we all stay active and do the things that we love for as long as possible? And the Stone Clinic and the work of the Stone Research Foundation makes that possible. So we got involved and decided we would be donors to this very worthy cause, and we're pleased to be involved. We urge all of you to think about how you can support Stone Research Foundation, and we are involved with other nonprofits. And one way to think about it is time, treasure, and talent. So if you have the treasure to donate, great. If you have time, I'm sure we can find ways to use your time. And if you have any talents that you think might be useful to the foundation, we'd really appreciate your help. So please join us in supporting the important work that the Stone Research Foundation continues to do.
Thank you, Mark. I appreciate it. Again, I hope that we don't have any more technical glitches here. Okay, great. Okay, with that, I would like to open this session now to our Q&A session, and I encourage you to answer any ask any questions that you may have, and both Dr. Stone and I would be happy to answer.
I'm going to pipe in first here with just a quick thank you to Mark and Laura and other board members and supporters over the years who have just really helped propel the science and information forward. It's a thrill to be able to care for each patient one at a time in the clinic, but it's really a thrill to know what I can do for them today and then what I will be able to do for them tomorrow. So where the research is going to. And that's the fun of practice. It's the fun of pushing science forward. It's the passion about what we all do. I notice there's another huge contributor to us on the line here, and Tom Turek has just been a core basic scientist for us for many years, and his contributions have been spectacular. And I see Darcy Roach on there too, who's inspired us to keep moving forward, and in his belief that we will get there and that we should never give up, which I'm a fan of. And I see many friends and familiar faces on the pictures here. So feel free to fire away if there's questions you have or interest in where this whole space is going to. It's a fabulous time in orthopedics.
I have a question. Can you hear me?
Yeah.
Hi, everyone. Question, are the regeneration procedures, do you need a certain amount of original cartilage still present? If you're bone on bone and there's very little to none left, is it still a suitable option, or is there a critical mass that's needed?
Yeah, our guideline, the question I get asked all the time is this knee available for a biologic solution, or is it over the edge and needs a bionic solution, which is what you're asking. And one of my guidelines is there enough joint space for me to put in a new meniscus or regrow the articular cartilage? If it's bone on bone, there's generally not enough space. And there we use a bionic solution where we use a robot and do a partial replacement or a full replacement. And that science has advanced tremendously as well. If there is joint space, then I can use your remaining articular cartilage and create a paste out of it and resurface that damaged area, add a meniscus, add a ligament, whatever's needed at the time.
Okay, thank you. So there
Yeah,
does seem to be, you need something there to kind of build on, it sounds
I
like.
need some space.
Yeah, you need some space. Okay.
Yeah, the fun parts of the bionics side of this, which is also a passion of ours to push forward, is that when the whole joint is worn out, we can now use uncemented implants where your body will grow into the porous undersurface of the implant. And by using robotics, we can make that so precise that we can fit them on now by removing much less bone than the old days and get you back to the sports you like to do on the uncemented knee replacement, including running. And if it's only worn out in one side of the joint, then we do a partial replacement. And that's been extremely effective for many people. We had a patient who ran across the United States and has 2 bilateral partial replacements. So the whole concept of, hey, can I get you back to sports? Can I have you drop dead age 100 playing the sport you love? Either biologically, we have so many more tools now, or bionically, we have more tools now. So there's no rest for the weary here anymore. Yeah.
I have a
Karen.
question.
Yeah.
Actually, 2 questions. One, does this pertain to the patella as well as the underside of the patella? That's question
It does.
one. Okay,
It
go ahead.
does. I just had the fun of looking back inside of a patella that I grafted years ago, a few weeks ago, and he had healed that surface over beautifully.
Hmm.
The patella is probably the hardest part of the whole knee, though, to get to regenerate.
Well, you don't have the space. You have to create a space, I take it, to get that to work.
Yeah, and it just doesn't have the great blood supply the way the rest of the bones in the knee joint have, but
Yeah.
it is doable.
Looking ahead, and I know you don't have a crystal ball, but when do you think this will be widespread and or readily available in your practice? And what's the state of play? You can't be the only person and group that is in pursuit of this objective. Could you give some color as to what that looks like as well?
Yeah, the one fun of having gray hair is that you've been around long enough to have seen the evolution of these spaces occur. And it's been an explosion of interest in cartilage regeneration in what I call the anabolic era of orthopedics, where we stimulate tissues to heal now rather than always just replacing them. So around the world, this is the hot topic in orthopedics. So we've been doing these regenerative procedures now for 30 years or more. And the question is, how do we make them better? And so, as you've heard about over the last 10 years, people have been injecting cells from every source or injecting growth factors. People have been going to Mexico and the Caymans and trying to get stem cells injected. Our belief, and I believe where the space is going to, is that within each one of you, no matter what your age, you have billions of stem cells. Otherwise, you wouldn't be here.
Mhm.
Yes, you have more when you're younger than when you're older, but you still have billions of them. And so a focus on our research going forward is, how do we recruit the billions of cells that you already have in your body to augment these regenerative processes rather than just injecting a few million from the outside? So we're taking a multi-pronged approach. One is where we add cells, as Monty mentioned earlier on, to our pacecraft technique. But other approaches where we stimulate your body to release your own stem cells and more of them to migrate to that site of injury.
And time frame?
Well, today, because we're doing it today in the clinic, getting better though, forever, because it's an evolutionary process. Here's where the future really will lie past what we're doing today. You've all heard that, you know that salamanders can regenerate their tail. You know that young children can regenerate the tips of their fingers. So we have within us the genetic ability to regrow tissues. And as we get better and better down these spaces of stimulation, we'll unlock some of these these DNA, these genetic components of regeneration. To timeline question, but the sooner we're funded, we can do all these studies that we want to do. Over abundance of studies I would do today if I were fully funded. But that said, we're pushing it forward today, and it will come in so many ways over the next few years.
in installments? Because you said you're doing in-clinic
Yep.
work today.
Yes.
But what I'm not clear on is if I or anybody else on this call said, I'd like to make an appointment to have the pace done on my knee,
Yes.
is that available today?
Yes. It is
Okay.
available today.
Thank you.
Yeah.
We have a question from Harry, I believe, next. And
Yeah. Hi, Dr. Stone. Remember me?
There.
Yeah.
Where
And
are
then
you here?
I think Shannon, the PT person.
Uh,
Yeah. What is it? I kind of regret. Yeah. I wish I had met you earlier because I got this 3 years ago. I got the total knee replacement at St. Francis Hospital, and I'm kind of disappointed with the results. They did a total knee replacement, and the surgery was successful initially, and then I was doing well in the PT, and then my urologist says, I think you may have the kidney stone in the right kidney. Drink a lot of water. So I suffered, what is it, a hyponatremia? It weakened me. I said, oh no. And then I went to my primary, and okay, say after that, so I went to the ER. They bolused me with saline. Try to make a long story short, he says, go back to your primary, get some sodium capsules, which I did. And then, so I have high blood pressure too. And he says, I asked him, do I still take my, diuretics? Yeah. And then I went back to the ER 2 weeks later, and the ER doctor says, your primary goofed. When you have low sodium, you never ever take the diuretics. It really messed up my PT outcome. And I went to see you. And I think, you x-rayed me, and I don't think you could help me because I already got the surgery. I still got the stiff knee. Excuse my language, but it sucks. I'm pissed off, but I wish I had met you earlier, you know. So yeah. So anyway, but it's good to hear, all the advances, in non-surgical, you know. So like in my case, I agree with everyone here. Avoid the surgery as long as you can, you know.
Well, it's good to hear. I'm sorry for all the trouble you went through, but there's two sides to that. And the two sides to it are, if you need to be repaired, be repaired. Life is short. It's more enjoyable to play every day today. You never know how many days you got. On the other hand,
Yes.
if we can get better and better at stimulating tissue regeneration, at shutting down inflammation, at helping people do well with injections or other therapies, and that's a big win as well. I have to tell you that we have an enormous number of patients who come into the office each fall with these horrible looking x-rays. And they say, hey, doc, I'm not going to let you operate on me until those injections you gave me don't work anymore. And you give me an injection in the fall and I get through the ski season, and sure, my knee's not great, but it gets me through the sports I love. So we don't know if we're, the guilty side of this is we don't know if we're helping our patients destroy their knees more or if we're just helping keep them out of the operating room. But these injection therapies are truly getting better and better and better. And part of our research, as Mani mentioned, is how do we identify which factors are the most potent ones and which patients should get which factors. So we designed this incredibly cool study to answer those questions. And we're looking forward to getting that funded and moving that one forward, because I think people want to know, hey, which injection should I get? When should I get it? How often do I need it? Can it help me avoid surgery? So I'm sorry you went through that trouble. We all are aware that not every surgery will work out the way we want it to. And we have to be diligent about trying to figure out how to help each person in their own individual way.
Okay. Okay. So there's no way you can help me, right? I mean, because I already got the surgery.
No, that's not true. I mean, we're there. Come on back. We're happy to take a look and figure it out and see where there are ways to be helpful.
Okay. Okay. I'll think about that. Okay. Thank you.
Sure.
Next, we have a question from Karen. I'm mistaken.
Hello.
Hi.
Hi, Karen.
Am I unmuted? I guess so.
Yep, you're good. You're on.
I've been working with you long enough to remember when amniotic stem cells were approved by the FDA, and then they were no longer available because the FDA rescinded their approval. And then just yesterday, I was reading in the financial news about a company whose stock is about to go up because they're doing a study with over 500 patients on an injection therapy. It's called Renew. Are you familiar with that?
Yes.
What are your thoughts about Renew? And also, I guess N-Stride is one that's a hyaluronic acid.
Yeah.
I
So Karen, great to see you.
nice to see you.
So what she's referring to is that the fountain of youth is probably birth tissues for a whole host of reasons. One of the reasons the mother doesn't reject the fetus, even though the fetus is only half the mom, is that the cells, the stem cells there and other factors immunomodulate that response, meaning they shut down the rejection response. And nothing is growing faster than the fetus. And so the anabolic effect of the growth factors there and the cells there are a source that we want to take advantage of. And so years ago, we would add these birth tissues to our injections in order to augment that healing response. The FDA then came out and said, hey, listen, the sourcing and procurement of those cells isn't up to their normal GMP or normal manufacturing processes. Moms just donate them at the time of C-section, and they wanted to regulate that industry, and so they did. One of the companies that said, okay, they will take the birth tissues through a drug approval process is the company that Karen's referring to, and their product is called Renew. And I personally believe that it will be tremendously successful because we saw early on when we used birth tissues their potent effect. So stay tuned on that. I think many of the birth tissues will be used. It will be a very interesting source of potency for anabolic therapies, and we'll take advantage of it as well. It's one of the components in the study that we want to get funded to see if it can augment articular cartilage healing.
And again, on that note, to echo Dr. Stone, this will be one of the arms, Morton's jelly, which is more potent, in fact, that we'll be testing in our arms. So we will know based on our study which one will win the race, if you will, for this particular indication, not for everything, of course. Darcy, I believe you have a question.
Yeah, can you hear me?
Yes.
Okay. Kevin, you finished the first phase of the CIRM study. I think it was looking at combining the paste graft with an adhesive. Can you tell us anything about the specifics of the results?
Yeah, we know that one of the reasons paste grafting alone didn't take off as one of the more popular ways of regrowing cartilage is that, number one, it was technically difficult for surgeons to put it in and keep it in place. And number two, the adherence to the surrounding articular cartilage may have been somewhat variable. And so by adding a hydrogel to the paste, we can improve the handling characteristics of the paste itself, as well as the adhesion to the surrounding cartilage. Additionally, by adding additional cells to the paste, which Serum was particularly interested in, we believe we can augment the extracellular matrix, the production of articular cartilage by the cells that are there in the paste itself. And that was what we saw in the rabbit study and the goat study that we've completed. We now will move on to a horse study, both to prove it in a large animal model, as well as to help horses and dogs, because horses and dogs get the same joint injuries that humans do, and they're both loved and very valuable animals. And so if we can help on the veterinary side, it helps us also on the human side, and both animals benefit. So we're optimistic based on the serum results that we should move forward.
Well, as you recall, I've had some experience with that adhesion issue with you in the past, not personally, but watching it. So is there any specific measure of improved adhesion?
Yes, there are. So what we've done during this time is analyzed various hydrogels to determine which hydrogel bonds at what rate to the surrounding articular cartilage. And we're right in the middle of a benchtop study right now to select which hydrogel bonds the best. And some of them, in terms of handling characteristics, can bond in as fast as a minute, and others in 15 minutes, and others take time for the body to heal and grow into them. So this timing issue is critical because surgeons, as you know, more than a minute is an eternity in the operating room. So our ability to add a hydrogel to a paste that then adheres well to the surrounding surface is one part of our assessment tool of which one should we move forward with. I won't say which yet.
Okay, thank you.
Yeah.
Are there any other questions that I have missed so far, Debbie, from anyone? Yep, go ahead, Jan.
I got another one. So if the organic stuff isn't so much an option, if there's not much space left, what about the synthetic cartilages? I know there's some that are in clinical trials. I think some East Coast universities, supposedly much stronger than in human cartilage. But again, is synthetic an option? What's your opinion on the state of synthetic? And again, what's your, what state do you have to be in to be able to take advantage of it?
So synthetic comes in different forms. One can be a regeneration template. I was lucky to invent the first collagen regeneration template for regrowing the meniscus cartilage back in the 80s. So that is coming back on the market. The harder materials that replicate articular cartilage in one way or another generally have all failed. And the reason is that you take 2 to 3 million steps at up to 5 times your body weight each year in normal walking. And if the cartilage is soft enough to protect the surfaces, it's not durable enough to last. If it's hard enough to last, then it damages the opposing bone and articular cartilage that it's rubbing against. So none of the materials have found that sweet spot, that Goldilocks spot in between. And that's because cartilage is a dynamic material. It absorbs water, it extrudes water with compression and release. It's the way the body heals. It has charged molecules within it called glycosaminoglycans that attract water and then release water. The whole viscoelastic properties of it are nearly impossible to reproduce artificially. So I'm not confident of any of the artificial cartilage materials yet. Someday. I saw there was a question about does it work in shoulders? Yes, cartilage regeneration procedures can work in shoulders. We stayed away from the shoulder because it was extremely difficult structurally and anatomically to get a cartilage repair process within a shoulder. There are efforts around doing allograft or donor plugs in the shoulder, but there has not been a lot of success around the regenerative efforts in the shoulder itself.
I believe Paul Stevenson has a question too. Paul?
Yes, thank you. I was just a kind of reflection. Both my knees needed to be worked on. I had stem cell done in both knees. I decided to have the stem cells for my own body because I was a little nervous about autoimmune response and such. Because I also had something called ankylosing spondylitis, or it's like rheumatoid. So I didn't want to risk it. And honestly, it worked for a few years, but my left knee was such a mess that mechanically it started to overwhelm the stem cells. But I did get a lot of growth, I have to say that, and in my right knee. And so I finally went ahead a partial done in my left knee, and it was very much like you said, no glues or anything, just into the bone and such. And that's been pretty successful, enough so that I ran a Dipsy race this past spring. And now my right knee needs to be done. I'm going to do a partial as well. So I'm big fan of the partial procedure. But but I started down the alternative first just to see if it'd work. And I probably would have done it all over again. So, but I mean, it was more of a comment than a question.
Yeah.
So
I think that's very normal. I think most people are trying to see how much benefit can they get from injections and great rehab. And that's the key. If you combine great rehab with your injection therapy, your likelihood of doing well is much better. So don't miss out on the second half.
Okay.
I have a question in regards to the regeneration of tissue with platelet, the plasma-rich platelet or other biologics. If you say have a scarred Achilles that didn't heal properly, so talking about scarred tissue, is there any experience or anyone has comments or particularly you, Dr. Stone, a comment on hope for something like that where you could regenerate tissue
Yes.
with scarred tissue?
Yes. So one of the very exciting aspects to both PRP and various cell preparations is that they are not only immunomodulatory and anabolic and antibiotic, but they are also anti-fibrotic, meaning they inhibit scar formation. And more than 100 years ago, abdominal surgeons realized that scarring after abdominal surgery was a huge complication for them. And they were applying amniotic membranes and amniotic fluid to decrease abdominal scarring after abdominal surgery. So I think a potent area of research going forward here is taking that scarring that sometimes is unhelpful, meaning you need some scarring for healing initially, but when scarring gets out of control or becomes stiff and fibrotic, how do you break that down, or how do you stimulate a more normal collagen formation to occur? And I think the birth tissues and that whole line of thinking is a potent possibility for doing that.
Can you do that at your clinic?
We can do injection. We can use amniotic membranes, and some of the birth tissues under FDA approval. In some ways, we can't do it. So FDA does not approve injections right now for arthritis. They do approve membranes for wounds. So yes, there's sort of a mixture of ways in which you can do things. And I believe this is all going to open up very quickly anyhow. So yes, and yes, you can use PRP, which also has that antifibrotic component. So it's worth thinking through each individual problem and thinking about it. Achilles tendons in particular, they tend to scar quite a bit. If I see a person with a scarred, decreased motion in Achilles tendon, our first line of therapy is injections of PRP with physical therapy. So I think there's huge potential there.
Thank you.
I saw there was a question there about preventing atrophy. Let me address that one. So it's a fascinating story that we are passionate about pushing forward a study on. So here's the story. If I hit you with an axe, otherwise known as surgery, within 8 hours of surgery, you start this atrophy process in your leg if it's a knee surgery. And that's because you have a massive cortisol, the stress hormone, that's released at the time of surgery. And that stress hormone binds your muscle receptors and induces that muscle atrophy process within 8 hours of surgery. So one of the studies we're fascinated by that I started years ago but didn't have funding to complete was, could we add a preloading dose of an anabolic called testosterone before I did the surgery, block those muscle receptors and decrease the amount of atrophy that you might get. So we got approval and funding from one of our grateful patients to do the first 15 patients. And we're looking for approval to do the next 85 patients, funding to do the next 85 patients. We have approval. And the question will be, can we preload you before you go to surgery and decrease the amount of muscle atrophy that you get? Now, it's hugely important because if I ask patients a year after knee surgery, hey, what was the worst part about your surgery? They never mention the pain. They almost always say, oh, it took me a year to come back from the surgery, get all my muscle back, rebuild the power that I had. And for athletes, for people working, for military, for everybody, if we can decrease that amount of atrophy, we'll make a huge contribution to the world. And so we're, Monty and Vicky can talk to that, but from a funding point of view, that's the only thing holding us back. We think that that can be a real contribution to the world.
Yeah. I will leave that to Vicky to, of course, follow up. She will follow up after this webinar, and a copy of a recording will be, of course, provided to each participant. So Vicky Weiland will follow up with the specifics on that with each and every one of you, and feel free to reach out to her directly or to any of us, of course, with any of those questions that you may have. Dr. Stone, there's one more question that was submitted ahead of this webinar in regards to meniscus repair. I don't know if you have that or not in front of you, but I can read the question if you don't have it.
I do. So question is about suturing the meniscus cartilage, and what's happening in that space and what are we using. So the meniscus is a fibrous tissue inside your knee joint, and when torn, it rarely heals on its own. We're biased. Again, I mentioned earlier, you always need to know the bias of your surgeon. We're biased towards repairing those tears anytime we can, because we know that if you take out part of the meniscus, you lose that shock absorption, you get force concentration in the knee, and it leads to arthritis. And so if we can repair them, it's much better for the knee joint. Now, traditionally, the repair success was variable, and we've gotten better and better at both the techniques, at the suture material, because we have a little flat ribbon-like suture now that we can use that's much stronger than what we used before. And we now almost always add growth factors or stimulation to that meniscus repair, either by releasing the patient's own marrow or by adding PRP at a later date in order to augment repair. So I think meniscus surgery and meniscus repair is improving in their outcomes, and hopefully they'll be used much more often.
Thank you for this answer of my question. Thank you so much.
Sure.
Thank
you. There is a clinic question that is basically somebody's asking if the Stone Clinic is in Carlsbad, California, and if insurance is covered for the procedures. And I'll leave that up to you, Dr. Stone.
So we're only here in San Francisco. Here, the Research Foundation and the clinic are adjoined here in the city. And we help our patients build their insurance. So depending on what insurance you have determines their reimbursement, but it varies all over the board. So we try to stay out of that side of it.
Are there any last minute questions that I can take before I wrap this meeting? Okay, again.
Yep, I would just say thank you for attending, and Manuel, thank you too. As you can tell, we're passionate about this. I mean, it's such a fun space to contribute to. It helps people who are in need. It's satisfying seeing people like Will come back years later playing the sports they love. It's really ripe for making contributions to the world. So whatever your philanthropic interests are, if this is a space that appeals to you, we're very appreciative of that support, and you can know that we'll be diligent and passionate with your trust. And I'll turn it back over to Mani, but thanks so much for listening.
I'll echo you there. I just wanted to thank each and every one of you for attending. It means a lot to us and our team. Your enthusiasm, support, and I hope more support moving forward will enable us to propel forward faster and better. So again, thank you. Vicky Weiland, our development director, will follow up with you, and we will also make a recording of today's session available to everyone here, of course. And on that note, I would like to once again thank all of our collaborators, some of whom are present here, our board members, and everyone else who's basically an advocate for SRF. To really a shout out. We wouldn't be able to do this without all of you put together, so here in this team. So thank you again. And on that note, I look forward to seeing you again soon on our next Research Progress. Take care. Bye-bye.